specific length of the DNA which can code for single protein. This is called genes. So it means that the gene is a specific length that is present on the DNA surface. For example, human insulin that contain 51 amino acids. Now, each amino acid is coded by triplet codon sequence. So there are the 153 nucleotide code on the surface of the DNA are making one genes. What about the gene length may be longer or it may be shorter. It depends on the different type of the proteins. For example, the hemoglobin is a larger protein as compared to the insulin. So the gene length for the hemoglobin is longer as compared to insulin. If we talk about the protein synthesis process, it consists of the transcription and the translation. So during transcription process, double helix structure of the DNA will convert into single strand by breaking hydrogen bond between the different nucleotide. For example, the gene length, which is available here, so this will split from double helix structure into single strand. From these two strands, one strand will provide a template strand for making messenger RNA. Now, this region of the gene contains two regions. For example, the one region is called start codon or promoting region. So this promoter region, that contain a start codon. For example, the thymine, adenine, cytosine will make on messenger RNA, so AUG. So that is the start codon, while the opposite region to the promoter region is the terminator region. This terminating region or terminator region, they contain stop codon. This stop codon, for example, the adenine, thymine will make on messenger RNA, the uracil, adenine. So terminating region is a region of the DNA where RNA polymerase terminates the transcriptions. So the transcriptions region confined it between promoter region and the terminator region. So our transcriptions will start when the one strain of the DNA will provide a template strand. So the free nucleotide will remove their extra phosphate and get activated and arrange according to the complementary base sequence on the template strand. After arrangement of these nucleotides on the template strand, they make hydrogen bond with the complementary nucleotide. And after making hydrogen bond, so the enzymes, they start working from promoter region. So RNA polymerase enzyme, they move from the promoter region and they start making the phosphodiester bond between the different nucleotide, and it proceeds on the surface of the DNA from the 5' to the 3' region. For example, it's moving from 5' to the 3' region and make phosphodiester linkage. And it produces a copy of messenger RNA from the DNA. This process known as transcriptions. So making messenger RNA copy of the DNA segment, that is transcription. And this transcription is taking place in the nucleus. After the transcription, the double helix structure will restore from the two single strands of the DNA, while the messenger RNA head toward the cytoplasm from the nuclear pore. So this double helix structure will restore by rebinding the single strand of the DNA, while the messenger RNA starts moving toward the cytoplasm from nuclear pore. When this messenger RNA reaches to the cytoplasm, so in the cytoplasm, it combines to the ribosomal RNA. And this ribosomal RNA move from the promoter region. So this promoter regions contain start codon. Usually it is the AUG. So in a normal transcription, it is in the AUG and which produce a methionine amino acid, but we're going to start with another amino acid as a symbolic representations of one type of question. So to answer that question, so this type of a sequence will be more helpful for understanding the question. So that's going to move from 5' to the 3'. So ribosomal RNA at the one time can expose the two triplet codon sequence on the surface of messenger RNA. So the transfer RNA, which contain at one side one specific amino acid, while on opposite side, it contain the anticodon sequence. So there is the triplet anticodon sequence on the opposite side of the transfer RNA. So it will bind with the exposed triplet codon sequence on the surface of messenger RNA. And in the one time, if there are the two triplet codon sequence are available, so the two transfer RNA can bind with messenger RNA. When they will match their codon on messenger RNA and anticodon on transfer RNA, their sequence, so they will make hydrogen bond. After making hydrogen bond, they will leave their amino acid and the peptidyl transferase enzyme make peptidic bond between two amino acids by use of one ATP molecule. So in this process, so by making a one amino acid bond, peptidyl transferase enzyme will use one ATP molecule. And after that, the ribosomal RNA will proceed from 5' to the 3' directions, meaning it's moving from the left to the right. While the used transfer RNA will move from the left directions, while the new transfer RNA will arrive from the right directions. So in this procedure, so you can imagine like the one airplane is taking off while the other airplane is taking on. So in this way, this process will continue. So the used transfer RNA will move from the left directions, while the new transfer RNA will come from the right directions. And in this way, the new transfer RNA will mate its sequence with the nucleotide on the surface of the messenger RNA. And after mating this sequence, so this will leave its amino acid and peptidyl transferase enzyme make peptide bond and use of the ATP molecule, and the used will proceed. Now ribosomal RNA will proceed to the further to the right direction, and the new transfer RNA will come from the right directions. And this procedure will continue once again by the peptidyl transferase enzyme. So this peptidyl transferase enzyme will make peptidic bond continuously from the 5' to the 3' directions, and ribosomal RNA continuously proceeding from the 5' to the 3' directions until the stop codon will arrive. So this stop codon, that is the uracil adenine. This stop codon will terminate this translation process. And after these terminations, one polypeptide protein will stay in the cytoplasm while the messenger RNA will disintegrate. Now, this messenger RNA disintegration is more important. If messenger RNA will stay into the cytoplasm, so the continuously one type of the protein will be producing in the cytoplasm, and they become surplus and as a waste. And that's the use of energy. And to avoid this loss of energy, so this messenger RNA will integrate. While the protein, so which is produced as polypeptide chain, this polypeptide chain will proceed to the Golgi apparatus to convert into specialized protein. Now, in the Golgi apparatus, this protein will convert into secondary or tertiary or quaternary in structure. For example, some protein they produce the enzymes to converting into the tertiary structure. What about some type of proteins? They convert into globular form, they produce fibrous protein. So the variety of different types of the protein produced in the cytoplasm according to their requirements. And in this way, translations will stop. So as we summarize this whole process, so the first, the DNA, which contain a specific length of the gene, will convert from double helix structure into single helix by the use of the variety of the different enzymes. And they contain the two regions. One is promoting region and second is the terminator regions. And the one strain of the DNA will make a copy of messenger RNA. And that messenger RNA will produce by transcriptions. And messenger RNA come out from the nucleus to the cytoplasm. And in cytoplasm, this will produce a protein, for example, the alanine, leucine, and alanine. And we can understand by sequence by matching the chart of amino acid with messenger RNA codon sequence. So the examiner can give us the sequence of the gene or the DNA, and we are supposed to convert into the opposite sequence on messenger RNA and match this sequence from the table and identify the sequence of the protein. For example, so the one concept, whatever there is a sequence of the nucleotide on the DNA, there will be same sequence of nucleotide opposite on the messenger RNA. And this messenger RNA sequence will determine the sequence of amino acid in the protein structure. So it means that whatever there is a sequence of nucleotide inside the DNA, this will directly determine the sequence of amino acid in the protein, while the sequence of amino acid in the protein will determine the characteristic of the protein. So in this way, so we can determine like there's a specific characteristic of the protein and depend upon the sequence of nucleotide in the DNA structure. So here on the basis of this following information, we can proceed about a specific type of question. Hope you got idea about this question. In this question, examiner is giving us the whole sequence of a protein synthesis process. For example, in the nucleus, the DNA, this DNA will make messenger RNA. And that messenger RNA, so that process is known as transcription process. What about this transcribed messenger RNA will come out from the nucleus to the cytoplasm. In the cytoplasm, the messenger RNA will combine with ribosomal RNA and the transfer RNA. And after combining with the transfer RNA, so this will produce a polypeptide chain. And this polypeptide chain, after processing, it will convert into the protein product like enzymes or the different product. So we're going to see about the different type of a question. For example, if there's a sequence of the DNA, that is means cytosine, guanine, cytosine, and this will produce on the messenger RNA the opposite sequence like guanine, cytosine, guanine, and that's going to proceed to the cytoplasm, and the opposite sequence on the transfer RNA, like anticodon sequence, will be the cytosine, guanine, cytosine. So you can see like on the transfer RNA, the sequence is reflecting on the sequence which is present on the DNA. So in this way, we can find answer. Examiner can ask us the questions to identify the different parts. For example, the A and B and C and D, we are supposed to identify these parts. So the A will be transcriptions, while the B is our transfer RNA, and the D is anticodon sequence on the one side of the transfer RNA, while the C is our ribosomal RNA. So in this way, this type of questions might possible. Examiner can also ask the questions to explain this process. So here, for example, this is a proceeding of the translation and state the sequence of the base at the J. So what is the sequence of the base at the J? For example, we're going to proceed from the left directions from 5' to the 3'. So the first is coming guanine, 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 and opposite sequence is the CCC. And second is going to match with the adenine to the uracil. And the third sequence is coming from AGC, while the fourth sequence, it's supposed to be GUC, and opposite of guanine, uracil, and cytosine, obviously it will come to the cytosine, adenine, guanine. So in this type of questions might possible, and you should be able to identify the sequence on the transfer RNA like anticodon sequence by watching the sequence of codon sequence on messenger RNA or the DNA. We're going to proceed to the second type of question here. Now, in these type of questions, the diagram shows the nucleotide sequence of small sections of the gene, which is transcribed. Now, this small section of the gene has been transcribed into the protein. So examiner has given us the table, and in this table, there are messenger RNA codon sequence are available. While in opposite, there is amino acids are given there. So what is the order of the 4 amino acids in the polypeptide translated from the small sections of the genes? Now, in this way, the messenger RNA sequence has been given there. So we are supposed to convert from DNA sequence to messenger RNA sequence. That's a similar type of question. Now, in this way, I converted, and this is the codon sequence into the triplet codon, and this triplet codon will produce on messenger RNA the opposite sequence. For example, the cytosine, guanine, cytosine, what example we have studied before, it's opposite to the guanine, cytosine, guanine. So you are supposed to convert the sequence on the messenger RNA, and this messenger RNA sequence, and you're supposed to make it by matching from the table. For example, so guanine, cytosine, guanine, and that is equal to alanine. Once again, this alanine is repeating, and the third one is coming uracil, guanine, cytosine, and that is equal to cysteine amino acid. Now, in this way, the sequence is coming, the alanine, alanine, cysteine, and alanine amino acid sequence. A will be the correct answer for this question. So in a similar way, we're going to see another type of question here. So the diagram shows the nucleotide sequence. This is coming thymine, thymine, and cytosine. So that's a similar type of a question. This table has been given. So we are supposed to identify first the messenger RNA sequence, opposite to like thymine, opposite adenine, adenine, guanine, and that's adenine, adenine, guanine. And in this way, we shall make an opposite sequence on messenger RNA. And then with the help of this messenger RNA sequence, we shall identify the sequence from the table. For example, the adenine, adenine, guanine is coming opposite to the lysine. And in the same way, we have on the lysine, and glycine and cysteine. So that's the coming of that, the sequence. For example, first is coming the lysine, and second once again is the lysine, and next we have glycine, and once again we have lysine. So this type of the sequence are there, so we can identify the answer. So by converting first into the messenger RNA, and from messenger RNA in the table, the sequence will be given, and we're supposed to match the sequence and find our answer. We're going to proceed to the next question here. So this style of question is a bit critical questions, but it's very simple to identify. The part of amino acid sequence in normal and sickle cell hemoglobin are shown normal hemoglobin, sickle cell hemoglobin, that's given there. So now in this way, a possible messenger RNA codon for this amino acids are shown there. So these possible amino acid codon, these are mentions. So which transfer RNA molecule is not involved in formation of this part of the sickle cell hemoglobin? So we are provided the 4 different type of the transfer RNA anticodon sequence. We are supposed to convert this anticodon sequence into the codon sequence on messenger RNA. And we're supposed to match this codon sequence with the given sequence, and we shall be able to identify which is not involved in making this 40 amino acid sequence of sickle cell. So we're going to see that, for example, first is A, we can convert it into the opposite sequence. So that's coming of cytosine, adenine, uracil, and that's opposite sequence is coming guanine, uracil, adenine. Second one is the B for the guanine, adenine. Adenine, that's coming opposite sequence, and that's for our C, adenine, cytosine, uracil, opposite to the uracil, guanine, adenine, and then we'll proceed with the G. This type of sequence we're supposed to write by pencil, and after that, we're going to match this sequence to the available sequence in the question. For example, for A, it's going to match with that, mean this is available, GUA is available. What about for the B? Trace it. It's also available here. While in the C, that's also available. If we check on the D, so have a glance on that. So no need to have a glance. Why? Because we already identify if the 3 are available, save your time, and the D will be your answer. So we need to be very tactful during the examinations. So due to the deficiency of time, we're going to proceed to the next question here. So the diagram show the stages in the production of part of the polypeptide chain. For example, the DNA nucleotide sequence template strain, these are given there. This template strain will produce messenger RNA sequence, and this messenger RNA will produce amino acid sequence. So which feature of the triplet codon is illustrated by informations given there? For example, you can observe, for example, we have leucine is going to produce from 2 different type of the triplet codon sequence. So we can come to the point here. So these are 2 leucines going to produce 2 different type of codon sequence. So we can say an amino acid can be coded for by more than 1 triplet codon. So that will be our correct answer here. Second, we're going to proceed to the last question here. That is this type of structured questions might possible. For example, a table 5.1 shows the triplet of the bases on the template of a polypeptide of the DNA for some amino acids. Now, in this way, these are the given there in the table 5.1, amino acid sequence are there, and the DNA triplet codon are being mentioned. While in the diagram and figure 5.1 shows the base sequence in the DNA and messenger RNA for the first 7 amino acid of the beta chain of a hemoglobin. Now, in this way, there are some fill in the blank spaces are there, means supposed to fill that with a reference of these other sequence. For example, the first one is our cytosine, adenine. Cytosine will help us to identify this opposite sequence, guanine, thymine, guanine. We need to be very careful. For example, if you're going to transfer this sequence from messenger RNA to DNA, we can apply the thymine. while this thymine is absent in messenger RNA, and we can write an opposite sequence from thymine to the uracil. So for example, there is another sequence on the DNA is based in thymine, guanine, adenine, and here opposite sequence of thymine will be adenine, guanine will be cytosine, while in adenine, here we need to be very careful. We're going to move from DNA to the messenger RNA, so there will be uracil. What about the third sequence? We need to identify the specific amino acid with the reference of the triplet codon sequence on the DNA. For example, the space is available, and in this spaces, the leucine is coming. I got this answer from the DNA sequence GAC. So we need to identify where it locates the GAC. So that one is a sequence. For example, GAC is locating on this point here. And this GSC is opposite to that's a leucine. So we can trace our answer in this way. So with the reference of the DNA sequence, you can identify the messenger RNA sequence. From the messenger RNA to DNA, examiner can ask, or with the reference of the DNA sequence, you are supposed to identify the sequence on the of the different amino acid with the reference of this table. So you are supposed to do the practice regarding that, and it will be more helpful for you. So we are going to complete this lesson. So thank you very much for watching. Hope it will be helpful for you.